Hi, everybody. I'm excited to welcome Dr. Bill Van Bonn and Frank Oliaro of Shed Aquarium in Chicago. Thank you both for being here.
Thank
You're
you.
welcome. Thanks for having us.
They recently published a paper as part of the MDPI Oceans Special Issue on Marine Mammal Health. And their paper is called Whale-Associated Microbial Communities Remain Remarkably Stable Despite Massive Water Community Disruption in a Managed Artificial Marine Environment. It's a complex title, so I want to break it down with them today. Let's start with you, Dr. Bill. So many things influence an animal's health. We're familiar with how bacteria can cause disease or how a healthy diet is important in keeping an animal healthy, but people might not think about how the water around an aquatic animal can play a role in their health. Can you tell us about how water and the tiny life inside of it can influence an animal's health?
Sure. What we're discovering is that the microbes, the unseen living things that share the environments with animals, particularly the aquatic environment, the vast majority of them, if not all of them, are in some way helpful to the animal. They are training the animal's immune system to deal with microbial life. Microbes have been around on the planet for billions of years, and of course, most animals much less time than that. So they've got to figure out ways to coexist with those microbes, and so that we tend to think of microbes only in a sense of causing a problem. But again, what we're learning is that most of the time they're actually helping the animals, and we need to really better understand that because our influence on the environment is influencing those microbes as well as the animals that live in it.
That's a great point. And so the microbes are everywhere around us. We have we kind of have a view of them of only causing problems, but there's really a lot for us to learn. And you all learned an important piece of science from this research. So Frank, you investigated how an animal's microbes changed when you take a large amount of water and exchange it in the system. And to do that, you looked at the microbes' genes. Can you tell us how exactly you study microbial genes?
Absolutely. So you can think of, plenty of ways to grow microbes, and that's traditionally how a lot of discovery had occurred with different bacteria in the past. But there's a real limitation because you need to know exactly what nutrients to provide and how to grow those organisms in the lab. So to be able to characterize an entire community of microbes, we use their genes as sort of markers to look at to say, okay, well, this bacteria is present in this community or not. And the way that we do that is by using molecular techniques like PCR and DNA sequencing. So similar to a test, for example, for COVID, which most people have heard of, getting a PCR test, we can use a PCR test to not only target one specific organism like a COVID test, but we can use PCR to target many organisms at the same time. And so that's what we do in the case of microbiome sequencing, where we use the PCR to basically find those genes of any of the bacteria in that particular environment. And then we use the sequencing technology to read all of those genes and tell us which ones they are. So we can compare the known sequence genes from bacteria to the unknown ones that we find in our samples, and then make some sort of a plot or use that data to look at what types of organisms are there.
That's a massive amount of data that you all are collecting. That's amazing. And using techniques that are really applicable to us in everyday life. Thanks for that connection there to COVID tests. So Dr. Bill, what was the most surprising thing you found in this study?
Well, it wasn't surprising, but it's still astounding. We expected essentially the findings that we documented, which was that despite the massive change in the community of organisms in the water because of a water change, massive water change. The communities that were associated with the whales did not change. They changed very little. And what that suggests is that there are some selected partners, if you will, that the whales have, that they allow to colonize them and to thrive there. And again, we think those are very likely organisms that are providing some benefit to the animal. We know of a number of well-discovered or well-studied microbe-host relationships, even in aquatic environments, like the bobtail squid has a light organ, the flashlight fish has a light organ. Those organs are actually tissues of the host animal that are colonized by one specific type of bacteria, that bioluminescence, that photoluminescence. And so these relationships are recognized, but how they are maintained is still somewhat of a mystery. And certainly for something like a whale, we still just really do not understand that. So this again leads to the better we understand all of the living things in an environment and how they interrelate, who's there, and not just who's there, but what are they doing? What functions are they providing? The better we're able to take care of those systems and animals.
That's super interesting. It is exactly what you said. It's astounding. It might not be. It's kind of what we expect, that microbes travel along with animals, but just to see it confirmed was a really interesting part of the research. So Frank, what's the most important piece of information that you want people to know about this work?
Well, I think one of the really important things is that not only, as Dr. Bill was just saying, that we here at Shed do everything in our capacity to take the best care of the animals as we can. So anything we can do to learn more about their environment, to learn more about how we can optimize the environment for their health is really important. But also, I think it's really important to note the power of aquaria and ex situ research, where we might not have opportunities to study, for example, the blowhole microbial community of a beluga whale in such frequency and resolution that we can here in the aquarium. For example, a researcher in the field might be able to take a quick swab of one animal and that's it, or a couple animals here and there. So having the capability to be able to obtain these types of samples is really powerful for not only our community, but also for conservation research that goes on in the field, that potentially we could advise on, that samples that wouldn't be able to be collected in that type of a situation. And so we also are expanding not only to just studying cetacean microbiomes, but also, we're doing a lot of work with pond turtles who are endangered up in the Pacific Northwest, and looking at their microbial communities and how we can maybe use Head Start programs in better ways to help their populations survive and bolster their numbers. And other situations where we do similar work using molecular techniques to look at, for example, the diet of an animal and how that relates to its microbiome or how that relates to its health. So having the capacity of both the laboratory capability as well as the opportunities to be able to perform some of these studies with the platform that an aquarium provides us is really, really valuable.
So you both touched on health in your answers there, and that's obviously a very vital part of the work that you do. And and in this research, so you collected samples of breath, feces, and skin swabs to study the microbial communities. And those are all samples that are a routine part of veterinary care. And Dr. Bill, can you tell us a little bit more about how you use the information that you found in the study, how that might lead to ways that you can improve caring for animal health?
Sure, really, it comes again down to just understanding those communities, the communities of organisms that are present, and what functions they're providing. What are they doing? Because we know that the human influence or changes to an environment will influence those communities. And so we need to understand what happens so that we can make good choices. For instance, we know that indiscriminate or overuse of antibiotic can wreak havoc on these microbial communities. That's seen in human medicine. It's very well recognized, and we're recognizing it in veterinary medicine as well. And it's important to understand it at the level of function, because many bacteria have the same function. So it's not only what organisms are there, but it's what functions are they performing, because it's the functions that are important, because there are redundant organisms who can take over if others are in trouble. But the better we understand it, the better we can promote it, promote an optimal condition for the health of the system, the entire system, right? And it's really about balance. So way back to originally, we think microbes tend to get a bad name. We hear about them when they're causing a problem. We hear about the viruses and the bacteria that cause challenges for us. But just the reminder here is that the majority are not causing trouble, and they're actually helpful. And in fact, viruses are all parasites. They have to live on a host. Many of their hosts are bacteria. And so, understanding the change in an environment, the influence that has on viral survival, can create downstream cascades of effects and allow certain bacteria that we may or may not want to happen bloom. So there's a lot of interest in really understanding this at that level.
Yeah, thank you for that. The understanding is the first really key piece of building the tools in your toolbox to be able to help. And so Frank, last question. What advice do you have for viewers who might want to do this kind of work that you do? They see this publication that comes out, and they might want to get involved in a similar way.
That's a great question. I would say it's extremely valuable information that you can gain out of learning how to sort of harness these techniques. And one of the things I love about doing molecular biology type research is that it's so transferable to so many different questions. And the one thing I would say is that even though it's extremely powerful, it's also extremely prone to things like contamination. And so being very cautious and being very careful in standardizing techniques and collecting all the appropriate metadata that goes along with the samples is really crucial to being able to make any interpretations out of the data, because you can generate just massive amounts of data using these sequencing techniques, and then sort of whittling through it all becomes a little more challenging. But I would say that gaining more knowledge about how to utilize these techniques is, I think, going to become more and more powerful as we get better sequencing technologies, which has really just expanded greatly in the past decade or so. And so now I think we're trying to catch up a little more with some of the education part of it. And so learning a lot of lab techniques, learning a lot of especially computing techniques are really, really important, because, like I said, interpreting these data can be challenging, and there's lots of it. And so whittling through that using a lot of bioinformatic techniques is really important. So there's definitely a lot of promise in learning more about those techniques as well.
Thanks for that. And thanks to you both for sharing your work, Bill Van Bonn and Frank Oliaro from Shed Aquarium. You can read this entire open access article at the link below. And thank you all both for joining me for the interview.
Oh, you're very welcome. Thanks for having us, Claire.